Welcome to TNPN Expert Education. In this video, we are going to see about plant tissue culture concepts, history and scope. Plant tissue culture. The term plant tissue culture broadly refers to the in vitro cultivation of the plants, seeds, plant pods on nutrient media under aseptic conditions. In this plant tissue culture, the complete plants are grown under the laboratory condition. So, the whole plant actually raised from the plant pods or seeds. Plant tissue culture techniques. See, this is the original plant. From this original plant, we have to collect the explants. So, explants are the part of the plant used for the culture system. See this picture, shoot, root, hypocotyle, leaf disc are used as the explant. Any part of the plant used for the culture system is called the explant. So from the explant, the callus was formed. From the callus, the shoots arise, then roots are formed. At last, the complete plantlets are formed. So this is about the tissue culture techniques. And what is the need for plant tissue culture? The needs are obtaining disease-free plants. So, by the conventional propagation, we are facing many problems like bacterial and fungal infections. To go through this, when obtaining disease-free plants, the tissue culture technique is very useful. And then second point is rapid propagation. Third one is somatic hybridization. The hybrids are produced by somatic hybridization method. Here the cells are fused together, the somatic cells are fused together to form the new hybrid plants. Genetic improvement. For the genetic improvement, the golden rice production are very suitable example, and the next one is androgenic and gynogenic haploids. Haploids are homozygous plant production. And the concepts of plant tissue culture. The totipotency. Actually, totipotency is the basic principle of the plant tissue culture. The totipotency means the potential of the plant cell to regenerate into the whole plant. So, each and every part of the plant cell have the potentiality. That's why the tissue culture techniques are achieved here. Actually, the plants undergo the two processes. One is de-differentiation and another one is redifferentiation. First, we are going to see about the de-differentiation. That means from the explant. I have already told that explant means any part of the plant. So, from the explant, the cells undergo the further divisions. It will form the callus tissue. Callus means unorganized group of parenchyma tissues. So, from the explant, the callus was formed here. This process actually called de-differentiation. And the next process is redifferentiation. Re means again. So, again the callus undergo the further division. It will form the whole plant with the shoot and root. So, this process actually called redifferentiation. So, this is the basic principle of the plant tissue culture. Okay, we are going to see about the history. In 1902, Gottlieb Haberland, just to see the picture, Gottlieb Haberland, he developed the concept of the in vitro cell culture and cellular totipotency. He made the first attempt of the tissue culture using the monocots plants. Hence, he is known as the father of plant tissue culture. In 1901, Thomas Hunt Morgan Penicillin, who coined the term totipotency in his book Regeneration. Actually, he coined the term totipotency. Morgan, simply T.H. Morgan. 1904, first attempt in embryo culture of selected crucifers by Hanning. In 1922, in vitro culture of the root tips, that means root culture by Robinson. 1957, discovery that the root of shoot formation in culture depends on the axin and cytokinin ratio by Sugan Miller. Actually, the growth regulators, that means axin and cytokinin, play a very important role in tissue culture process. It was first discovered by Sugan Miller. 1959, Gottret, he is the father of plant tissue culture. He published the first handbook of plant tissue culture. 1962, development of the MS medium. Actually, this is a milestone in tissue culture process. MS means Murashigan Suk. MS medium is the common medium used for all the tissue culture technique like anthoculture, embryo culture, whatever it may be. But the MS medium is the common medium used for all the tissue culture techniques. In 1964, first haploid plants. The haploid plants are produced from Datura through the process of the androgenesis by Guha and Maheshwari. They are the Indian scientists who first introduced this haploid production. So this is a very important thing. You have to remind that the androgenesis process first introduced by Guha and Maheshwari in Datura plant. In 1973, cytokinin is found to be capable of breaking dormancy in gerberas. In 1978, somatic hybridization of tomato and potato resulting pomato by matures. Actually, this pomato plants have both tomato and potato plant. This pomato actually produced by somatic hybridization technique. In 1978, industrial skull fermentation of plant cells for production of chiconin. Chiconin is one of the secondary metabolite. It is used in the pharmaceutical product production. It is first introduced by Tabata. In 1981, introduction of the term somoclonal variation means the variation observed in the tissue culture plants. Actually, this tissue culture process achieved by somatic cells. So, this is called somoclonal variation. The term actually introduced by Lokin and Schoolcraft. Actually, this is one of the important thing. You have to remind that who first introduced the term somoclonal variation. It is usually asked in the examination, so don't forget, by Lokin and Schoolcraft. In 1985, infection and transformation of leaf disc with Agrobacterium tumefaciens and regeneration of transformed plants by Horsch. So, Agrobacterium mediated method of gene transfer nowadays used widely in tissue culture and transgenic plant production. In 1985, development of disarmed TA plasmid vector system for plant transformation by Fraley. In 1985, development of binary vector system for plant transformation. In 1985, gene transfer in protoplast of dicot and monocot plants by electroporation method. Electroporation is one of the direct gene transfer method. So next we are going to see about scope and importance of the tissue culture. In a relatively short time and space, a large number of plantlets can be produced starting from the single egg plant. So this is one of the important thing. Within the short duration, we are producing large number of plantlets here. Taking an egg plant doesn't usually destroy the mother plant. So rare and endangered plants can be cloned very safely. It is easy to select the desirable traits directly from the culture setup, that means the in vitro setup, thereby decreasing the amount of space required for field trials. Once established, a plant tissue culture line can give a continuous supply of the young plants throughout the year. So this is one of the important points here. The time required is much shortened for species that have long generation time, low level of seed production, or seeds that readily do not germinate, like orchid plants, the seeds are not readily do not germinate. So for that crops, the rapid propagation also possible through by the tissue culture technique. In vitro growing plants usually free from the bacterial and fungal diseases. So this facilitates the movement of the plant across the international boundaries. For example, tissue culture, bananas, they are free from the viral infection. Plant tissue banks, it can be frozen and then regenerated through the tissue culture techniques. So production of the exact copies of the plants that produce particularly good flowers, fruits, or have other desirable traits also. I hope this video may useful for you. Please like, share, comment and subscribe for more videos. Thank you.